Let's take a look at microbial metabolism. It's a pretty long chapter, has a lot of information in it, but it will be the last topic that's included with your Unit 2, and that will be on the upcoming exam too. The big picture, well, metabolism, it's a big word. It's the sum total of all reactions taking place in a cell. And that includes those reactions that build up energy, those reactions that release energy. So it's the sum total. Taking a look at how we can break this down into a little bit smaller categories. You can divide metabolism up into those reactions that release energy. That would be catabolic reactions. So catabolism means that we're starting out with a larger molecule and we're going to break it down into simpler ones and we release energy. So we're going downhill as far as energy is concerned because these molecules have less energy than the starting compounds. And energy is released in catabolic reactions. Anabolic reactions go the opposite way. We're starting out with smaller molecules that have less energy. We have to add extra energy into the system, and then we're building up larger molecules, say proteins, for example, that have, or the resulting molecules have more energy than the starting compounds. These reactions are typically coupled, so some reactions here, they go downhill on the energy scale, those catabolic reactions, and they deliver the energy that's typically stored as ATP, and they provide the energy for anabolic pathways. When it comes to bacteria, we actually a lot of times take advantage of their metabolism, and we use them to make cheese and wine and for bread. So a lot of times we find the metabolism of bacteria rather beneficial. But of course there are some pathogenic pathways too that create toxic substances. Also, sometimes microbial metabolism can cause disease. Here are some examples where you can see that we're utilizing microbial metabolism for our benefit. Some examples here, those microorganisms that make antibiotics such as penicillin or erythromycin. We can also use microbes to help us make vaccines and vitamins, sometimes also enzymes that we could use. So there are quite a few positive things that we get out of microbial metabolism, and it's important to note that because a lot of times we think that microbes, their metabolism just makes us sick or spoils our food. Anyway, back to the reactions. So one more time, catabolic versus anabolic reactions. Catabolic reactions, they go downhill on the energy on an energy scale. That means we're breaking down larger macromolecules into smaller ones and we are releasing energy. And then anabolism, we're using that energy that came from catabolic reactions and make larger, more energy containing macromolecules. There are two more terms. When you're releasing energy, means energy out, X, it's exergonic. That's another term you need to know. And when you're putting energy into the system, well, these reactions are endergonic. So anabolic reactions require energy. That means they're endergonic, and catabolic reactions are releasing energy. That means they are exergonic. So here, it's a nice summary slide that takes a look at that. If we are breaking down glucose into CO2 and water, well, that's a catabolic reaction that releases energy. That energy can be stored in form of ATP, and then that ATP can be used to, let's say, make proteins from amino acids, which requires a lot of energy and therefore is called an anabolic reaction. In metabolic pathways, they are sequences of enzyme reactions in a cell, so there's one thing leading to another, and typically in cells, metabolic pathways, it can be pretty lengthy, and there's many enzymes involved that are doing the various steps. Let's take a look at enzymes. For the most part, they're proteins, and so therefore they're encoded by genes. And the way they work, there are basically two models out there. One of them is more static. It's more like this key and lock principle, where the molecules are sort of, they have to fit together, and then one enzyme catalyzes one specific reaction. The other model is called the induced fit model, and there the collision of the enzyme and the substrate will create the binding pocket that's necessary for the reaction to occur. Either way, you need to have some interaction of the substrate with the enzyme, and that leads us to the collision theory. That means these two molecules, they have to bump into one another, and since molecules have more speed and more energy when they're hotter, that means when you're heating something up, your chances of a collision, a forceful collision that has enough strength to produce a reaction, the chances are much higher when the temperature is higher. So that means typically enzymatic reactions will be faster if you have more heat in the system. Now, proteins, they denature at a certain point, and so you can only take it so far. But anyway, back to sort of this, the whole big picture here. For any enzyme reaction, you need to first overcome a hill of energy, and that's called activation energy. So you need to first go a little bit uphill to make these molecules even want to react. And then once they overcome this activation energy, then the reaction will proceed from there on. The rate of the reaction is how many molecules of product you can produce in a certain amount of time. And of course, the reaction rate can be increased by heating up the system, so increasing the temperature. Again, you can't overdo it because enzymes are proteins for the most part, and they will denature if it gets too hot. But sometimes also concentration of certain substances can increase the reaction rate, or sometimes pressure or other parameters. So, more terminology on enzymes. In general, enzymes are considered catalysts. They speed up chemical reactions without being used up or altered in the reaction. So that means they just increase the rate of something occurring. These reactions would occur naturally, but it would take way longer for these reactions to occur. So catalysts, they speed up chemical reactions. And enzymes, of course, are biological catalysts. They need a substrate in order to produce a product. So let's take a look at this. Here's sort of this classical image. You're starting out with your reactants right here. You have reactants A and B. And in the end, you want to produce products separate A and B. Now, normally, without an enzyme, you would have to go all the way up here to get this reaction to go. So reactions don't go this way straight down. They need to first go uphill and overcome this hill of activation energy before they react and go downhill on the energy level and produce the products. This is relatively unlikely to occur, so that would have to be a very forceful collision of these molecules in order to overcome this hill of energy and then produce the products. It's not impossible, but it's usually very unlikely. And it protects ourselves from having random reactions that we didn't want in the first place. Now, if you are having an enzyme, then you can lower the amount of activation energy. Now you only have to go to here, and that's much easier to do. So now you can take your reactants gently uphill a little bit, overcome this hill of activation energy, and then produce your products down here. So the big thing about enzymes is that they lower the amount of activation energy. You can see that the red bar here, this energy amount, is much less than the energy that you would need in order to get the reaction going without an enzyme. So this here is without the enzyme, this huge amount of energy from here to here. And with enzyme, we only go from here to here. So plus these enzymes are very specific, so only the reactions that you want to have you will catalyze with a specific enzyme, and then you get your products A and B fairly easily. So the way that enzymes work as catalysts is that they will interact with the substrate, and then they first form an enzyme substrate complex, and that is when they are overcoming this hill of activation energy, and then the products will be released from the enzyme, and the enzyme is recycled and will interact with another substrate molecule. So here's a diagram that shows that. Here is a substrate molecule in red, and it's going to fit into this binding pocket on the active site, and then they form this enzyme substrate complex. And then this is the place where all of these new bonds are being formed or broken. So this substrate molecule is modified, and then the products will be released, and the enzyme is recycled and will grab onto the next substrate molecule. So here it's maybe more realistic, kind of more space-filling kind of model of the same thing. And the enzyme specificity and efficiency, it's amazing actually. So enzymes are highly specific for their substrate, and they are very efficient. We call that a turnover rate, and it's pretty staggering. That's the number of substrate molecules an enzyme can convert to a product per second. And it can be really high. Take a look at this. When this enzyme is really going, it can be up to 10,000 product molecules per second of this enzyme working on those substrates. So anywhere from 1 to 10,000, but that's a pretty large number. So here are a few comments about naming of enzymes. Generally, they end with ASE. So it's not always the case, but generally speaking, enzymes end with ASE. Here's some examples given: oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. So a couple exceptions that I can think of would be pepsin. That's a hydrolytic enzyme that works in the stomach, but in general, enzymes end with ASE, and that's when you know that it's an enzyme. Sometimes enzymes need partners. That could be cofactors or coenzymes. Let's take a look at that first. Usually a cofactor would be a metal ion, let's say a zinc or an iron or some other metal ion that's not a protein component. And a coenzyme would be an organic cofactor such as a vitamin or some sort of maybe NAD or FAD, those kind of coenzymes. They're larger, more organic kind of molecules that help the enzyme to do what it does. So when we put it all together, the apoenzyme, that's just the protein portion of the enzyme, then it may or may not need cofactors. There could be either a protein component, like an organic cofactor such as a vitamin, or it could be a non-protein component such as a metal ion like iron or zinc. When you put these two together, the apoenzyme plus any cofactors, we have the holoenzyme, the entire enzyme. So here's a diagram that shows you the apoenzyme here first. That's just the protein component. Sorry about that. That was my dog. Then here, the coenzyme, that could be, again, it could be a metal ion or a vitamin. So here we have the coenzyme embedded here now into the protein of part of the enzyme, and then here is the substrate that now fits into this binding pocket. Here is a list of a few of those coenzymes. NAD is very popular. It's nicotinamide adenine dinucleotide, so it is a dinucleotide. Then we have here a nicotinamide adenine dinucleotide phosphate or flavin adenine dinucleotide FAD, coenzyme IA. Those are very common coenzymes. Now here, factors that influence enzyme activity, the most important ones are listed right here. Temperature is a huge factor because of the collisions that enzymes and substrates have to go through. and they of course increase with temperature. pH is a big factor. The substrate concentration is a big factor, and then the presence of any inhibitors, whether competitive or non-competitive, but inhibitors